Welcome to our lesson on DNA and protein synthesis. The key questions for today are: what is the structure of DNA? How are proteins made? How do DNA mutations occur? How do DNA mutations change the protein made? What are proteins used for? The key words you will need to know are ribosomes, synthesis, polymer, mutation, enzyme. We have already learnt about the structure of DNA and the base pairings that occur. Task: describe the structure of DNA and state the names of the 4 bases. Extension: state the bases that form the complementary base pairs. DNA has a double helix structure. It consists of sugar phosphate strands and nucleotide base pairs. The bases are A, T, C, and G. A and T always bond as a complementary pair, and C and G always bond as a complementary pair. DNA is described as a polymer because it consists of repeating units. The DNA codes for the production of proteins. Proteins are made of a long string of amino acids. Specific proteins contain specific amino acids, and DNA bases code for this. 3 bases, called a codon, code for an amino acid. Each amino acid has more than one codon that codes for it. For example, the amino acid alanine has the codes GCA, GCC, GCG, GCT. Task: answer this AQA exam question. Proteins are made by ribosomes. During protein synthesis, a copy of the DNA code is made and transferred out of the nucleus and into the cytoplasm. It is here that the ribosome moves along the code, joining the amino acids together in the correct sequence. The amino acids are transported to the growing chain by carrier molecules. Protein synthesis requires energy. This energy is generated in the cell through respiration. Respiration takes place in the mitochondria. Extension: the cells in the pancreas make enzymes. Explain how the genetic material in the nucleus enables the enzymes to be made. The DNA in the nucleus provides the code for proteins to be made. A copy of the DNA is made and moves out of the nucleus and into the cytoplasm of the cell. Here, a ribosome, with the help of carrier molecules, builds up a strand of amino acids in the order dictated by the code. This strand of amino acids is a protein that folds into a specific shape to carry out its function as an enzyme. This picture shows a ribosome moving along the strand, reading the code and lining up amino acids to form a long strand or protein. The tRNA is the carrier molecule that brings the amino acids. If the DNA code is not correct due to a mutation, the wrong amino acid could be included, leading to a different protein being produced. The mutations in the DNA could be as small as a base substitution, just one base is incorrect. This does not always lead to a different amino acid, as some have many different codes for them. This type of mutation also only affects one amino acid in the protein chain, leaving the rest of the protein unchanged. A mutation involving the deletion or insertion of one or more bases can cause more drastic changes, as the whole code from that point onward is changed, meaning that many of the amino acids could be incorrect, resulting in a different protein being produced. Task: sickle cell anemia is an inherited disease caused by a mutation in the hemoglobin gene. Genes are small pieces of DNA. The DNA in a gene consists of a sequence of bases. The table shows part of the base sequence in the DNA of a normal hemoglobin gene and the same section in the sickle cell gene. A, C, G and T represent the different bases. Describe how the mutation causes a change in the shape of the hemoglobin protein molecule. 3 bases code for 1 amino acid. The middle code of CTC is now CAC because the T changed to A, so will be a different amino acid in the chain, so the protein will have a different shape due to a different sequence of amino acids. Extension. Syndrome H is an inherited condition. People with Syndrome H do not produce the enzyme IDUA. Figure 3 shows part of the gene coding for the enzyme IDUA. Strand K shows a mutation in the DNA, which has caused Syndrome H. The enzyme IDUA helps to break down a carbohydrate in the human body. The enzyme IDUA produced from strand K will not work. Explain how the mutation could cause the enzyme not to work. A mutation changes the codon CAG to TAG. This changes the amino acid that is coded for, which could change the protein. This means it forms a different shape, so the enzyme no longer fits the substrate. This could be a problem, as enzymes and hormones are proteins, and if they are the wrong protein, they may not carry out their function correctly. This is because once a protein is synthesized, it folds into a unique shape, and if it is not the correct shape, it cannot join with its specific substrate, and therefore cannot carry out its function. Not all parts of DNA code for proteins. The parts that do not code for proteins are called non-coding, and these can switch genes on or off. This means that a characteristic could be coded for by the DNA, but the way it is expressed could be altered by the non-coding part of DNA. For example, some parts of DNA that code for the production of enzymes could be switched off, resulting in the enzyme not being produced even though the code is present.